Okay, good afternoon. Thank you for joining us here this afternoon with the first of this semester's Westwood Lecture Series. We are very excited and anxious to hear today's lecture topic on the universal influenza vaccine from a distinguished colleague of ours. Professor Suresh Mittal is a distinguished professor of virology in our College of Veterinary Medicine, and he has been a leading expert in the country when it's topics related to vaccines during multiple episodes of health crisis in the country over the past decades. And he's a leading expert on this very timely topic, especially as we read in the news on a very frequent basis these days about all kinds of new health threats we face and wonder about solution sets. So I'm not gonna be eagerly awaiting the answer to your question, aspiration or reality. So again, welcome, Professor Mittal.
Okay. So the way I have this topic, because when people talk about universal influenza vaccine, it's so broad because there are 198 different types of influenza. And then within that subtype, there are so many different clades. So who is going to do all the testing with each and everything? So probably I like to have, rather than universal flu, because WHO, CDC, NIH, they come up with this term and they are, you can say, popularizing that term. So yes, can we able to get the vaccine which can work? What are the relevant viruses? I think that can work. At least what we are getting it, we are excited about that possibility. So when we talk about flu, many times, many of us or public in general may not consider it's a killer. So consider the cases which are occurring in people. I think it won't come there. So those illnesses in the world, more than 1 billion cases are occurring, and then more than 500,000 deaths worldwide. In the US, the death can vary from 5,000 to 50,000, and cases can be from 9,000 to 40,000, or 9 million to 40 million. So the cases are huge. Just looking at what has happened last season. So last season, if you look at the US map, the cases was everywhere, and total cases, I think, yeah, it won't go there. So 40 million cases overall, and the death about 28,000 in the US alone. So that season was a little bit, you can say, more severe season, but still that even our seasonal flu vaccine has to be better because providing protection. So when we talk about influenza, which family it belongs to, but more importantly, what is type of the genome it has. So the genome is segmented genome. So it has 8 different pieces of the genome, and each of those pieces represent one gene. And then what it causes that this different flu can be in one species, and those 2 flu, when they are replicating, then these pieces can sorted in a way that a new virus can be generated very easily. So in addition to that, there are some mutations which can occur every year because of the faulty polymerase the virus carries, as well as immune pressure, it's also there. So the structural protein, which are the basis of this typing of the virus, is hemagglutinin and neuraminidase. So hemagglutinin type in the birds, represented as HA, there are 18 of them. And then neuraminidase, NA, there are 11 of them. So you can combine all together, they will come up 198. So when we look at its cross-species transmission, these aquatic birds, they are the central stage because they have all different viruses, all 198 of them. So they are capable of giving these viruses directly to human or to poultry. From the poultry, it can come to the human directly, or from poultry, it can go to pig, and then from pig, it can come to human. Even to marine mammals, these virus can go, and from marine mammal, the virus can come to human. In addition, we know that the dog can get infected now. Previously, there was no virus which was in the dog. Now, we also know the cat can also get infected with influenza. It was not there, cat did not have any influenza virus. And then we know the horse is getting influenza for a long period of time, so the horse virus mutate a little bit and become a dog virus as well. And now we are dealing in the US that H5N1, which is a avian flu virus, can go to cattle. And from the dairy cattle, the virus is there in the milk. So there is a concern that milk supply can be contaminated, and then how we control these viruses. So when we look at the pandemic, the first flu pandemic which occurred more than a century ago, and coronavirus infection, you can say a few years ago, was also a big pandemic. But in that pandemic, if you look at the life expectancy, that graph, that is life expectancy of the US has dropped significantly just because of the influenza. And if you consider the total world population at that time was 1.8 billion, there were cases more than 500 million. Some people say close to billion. And then death are different, people are saying different way, but at least 20 to 50 million death has occurred. At that time, mode of transportation was not that much. Even then, the virus spread. So if the similar kind of virus can become pandemic, it can be a big deal. So if we look at initially, 3 pandemic was avian flu, that the 4th one, which was much milder in 2009, they call it swine flu, but it has those segment of the genome from 3 viruses, which has origin from human, pig, and avian species. The antigenic drift, they are the slow changes, slow mutations, what mutations are occurring in coronavirus, and with that one we calling them different variants. So those same changes are occurring in influenza every year at a faster pace. That's why every vaccine for seasonal flu every year, that has to be tailor-made to anticipate that which strain are going to be in our flu season. If we have a good match, then the vaccine will work better. And if there is a mismatch, then vaccine efficacy that particular year is going to be down. And there is no other way. So we also need a better vaccine for seasonal flu so that without predicting which strain are going to be around, we can have a vaccine which is going to be effective. So these are the viruses for seasonal flu, H1N1, and then H3N2, and then there are influenza B also, and there are 2 lineages, Victoria and Yamagata. And from the information I'm getting from the, you can say, publication, that because of COVID and because of the mask mandate and those things has happened, probably the Yamagata strain may be extinct. We haven't seen the cases of Yamagata strain in human since 2021. So it will be good news. We have to have a vaccine where seasonal flu only containing 3 viruses rather than quadrivalent vaccine. So we'll see if it is still around, because this virus was in human. So when those measures were there, the virus probably has disappeared. In addition to that, some of the significant avian viruses which are there, which can cause human infection, there are 3 of them. H5N1 causing almost 900 cases and almost 50% mortality in human. Then H7N9, and majority of those H5N9 cases has occurred in China and in that part of the world. Then less, you can say, virulent in human is H9N2, and out of 125 cases, there were 2 deaths. So there is a lot of concern going on currently with H5N1 virus, because if you look at overall in the world, that H5N1 cases has occurred in wild birds, and from wild birds has come to poultry, from poultry it has come to human, and also the marine mammals. And then in some of the slide we'll see that many of the wild mammal are also getting infected with H5N1 virus. So the virus is rampant in North America, in Central America, South America, Europe, Asia, part of Africa, but seems like that Australia seems to be getting spared. So this is the evolution of this H5N1 virus. It started in 1996 in Hong Kong, where initially I think the virus may be there in poultry for some period of time, but when human infections started coming up, then there was a huge concern why individuals are having infection, and not only having infection, many of them are dying. So since then, we thought the virus could disappear or become less virulent, but so far, after even, you can say, close to 30 years, the virus is still around and has multiple recombination event, and it has even wider its host range. So what next for this virus, we don't know. Is it going to become a pandemic virus? No idea. Or it is going to disappear? It can. So this virus is not a pandemic virus, the reason being that it is currently growing in the lung of the human, not in the upper respiratory tract. So once there are few changes in the virus, as soon as it can start growing in the upper respiratory tract, we have a big problem. Then it will become a pandemic virus. So these just to show you that how, you can say, beautiful for virus point of view, when they evolve, that looking at these different viruses, and these are the segment from different species, and how they get, you can say, get together. So if we look at these two get recombined and then creating a virus which is very different. So this virus, clade 2345b, was seen in Europe, in Spain, as well as in Finland, and causing a huge problem there. So this somehow, this similar virus came to the US, then going through multiple reassortant events, and one virus ended up in South America, in marine mammals, and another one was in the US, initially in the poultry, and from the poultry it also came to cattle. So currently when we look at the wild birds in the US, every state has positive birds, positive aquatic birds, or these migratory birds. So percentage can vary. So when we catch them and take out the samples, then that's the way it is. And even Hawaii has positive birds. So what has happened in there that all 50 states in the US has infection in poultry with H5N1 virus. From 2020 onward, 135 million birds has to be euthanized. And if you just look at current situation, that in the current situation, in 1 month, 12 million birds were euthanized. So it is a infection in the birds is still going on in the US, and it has been staying much longer than anybody thought about it. So I wanted to mention that a few years ago, I did not know even that information that Indiana is first in duck production, and then second in egg production, third in turkey meat production. So it is a significant poultry industry is a significant in Indiana, and that is contributing a lot to the economy of the Indiana. So when we are looking at, instead of poultry, all wildlife, excluding these wild birds, then there's a lot of different mammals, they are coming positive. Consider the wild cat, domestic cat, bear, and also marine mammals, then rabbit, hare, and then skunk, raccoon, and badger, and even opossum, even rodent. All of these animals are getting infection. So it seems like that virus is going to the multiple species. With that one, how it is going to evolve. So once it is evolving, then is it going to be much worse for human or for any other species? So similar thing, if we can say that one, that it is having so many linkage with birds, so many linkage with wild animal, and so many linkage even to domestic mammals. So where should we look for? That what changes this virus can have and what consequences of those changes is going to be. So the virus which came to the cattle and which H5N1, and that one, we know multiple event has occurred. So once that virus was in the cattle, so from the cattle it can go to the avian species, it can go to the wild animal, and from cattle it can also come to human. And from cattle farm it can go to the vehicle which are coming in, then those vehicle goes to another farm, then infection can be there. These animals go to their own farm, other animals are there, it can get infected, or any infected animal from this herd is traveling to another place, then infection will spread to that locality as well. So we know that 16 states has the infection, about more than 900 cases has occurred. And one thing is there, that milk production in those cows will go drastically down. The milk become yellowish in color, it will be thickened, and they will be having low-grade fever, mastitis kind of sign are there, respiratory signs, they are inconsistent, and amount of virus in the milk is the huge amount in the milk. Transmission, people think that the main transmission is contaminated milking machine. So I think exact mode of transmission is still questionable. The respiratory route, can they able to get infected? Animal to animal infection, do they require a huge amount of dose or a continuous exposure? Is there, because otherwise that many animal won't get infected. But all the animal in the herd, they don't get infected. So it seems like the spread is not that fast. The virus can change and then spread can be much, much faster. So since the virus is there in the cattle, it's bound to come to human. So overall, 67 confirmed cases has occurred in the US and leading to 1 death. So many cases or more cases has occurred in California compared to other states. But good point is that human-to-human transmission has not been seen in the US. But in other part also, human-to-human transmission was not there. So that is a good thing that at least the virus has not changed enough. So these are the definition from NIAID from NIH, that if a universal vaccine somebody prepare, it should be at least 75% effective and protect against group 1 and group 2 influenza viruses, and durable protection at least for a year and suitable for all age group. So they are just talking about human. They are not talking about animal. In this one, they did not include influenza B. So many questions arise that it should be against all influenza viruses or against the one which are relevant to human currently. Because for all influenza viruses, the work has to be done, a huge amount of work, before you can declare that vaccine as a universal vaccine. Then influenza B should be covered if it is for human. And immunity, it has to be sterile immunity or something else. The vaccine should prevent transmission. Because many times, if the transmission is not prevented, the infection cannot be stopped. For example, COVID vaccine, when we know now that COVID vaccine was not capable of preventing transmission. Even that was not able to even prevent infection. So people getting sick and they can replicate the virus, they can spread the virus. So they were only that COVID vaccine is protecting from the death. And then when effective criteria of the vaccine effectiveness, what are those criteria? The virus titer in the lung should be of 2 log down or 3 log down, or there should not be any clinical sign or anything else. And which species? I think they mentioned human, but I think other species are also important, depending upon these species that can we have the vaccine for those species also. So when we look at these different group 1 and group 2, then there are so many of these hemagglutinin are there. So the one which are circled, these are the one which cause infection in human. So influenza B is very different from group 1 and group 2, but there is a clearly antigenicity. These 2 groups are separate. So I think I prefer the broadly protected vaccine. So the system what we are using is a bovine adenovirus-based system. So that is sort of a unique system. I think nobody else is using that system. I started that system in Canada, and then those groups, they are using little bit of that. So we have moved that system to the point that it is a highly versatile system now. So nobody else has the know-how of this system. That is a good advantage. So these genome organizations similar to human adenovirus, then also it is not causing any infection. It's in host. In human, this virus cannot cause the disease. It does not replicate in human. So all our vaccine are replication defective vaccine. Receptor usage, these are the sialic acid receptor, influenza virus also using the same receptor. So mucosal vaccination we can do much better. So there is no pre-existing adenovirus antibodies in human which can neutralize this virus, and then there are multiple advantage. And I'm not presenting here, we just have done the work in the lab to find it out that does our virus bind to some blood receptor of human, because many other adenovirus, they do bind. So far, we have done multiple of those blood factors. So none of the blood factors seems to bind to our virus, and which is a good thing because some toxicity can be there because if the virus is binding to some of the blood factor. So when we talk about hemagglutinin, where the seasonal flu are already based, so in that kind of vaccine, what will be there? So here what they did for almost 10 years, different virus, H1N1 virus, they look at what are the modification in the hemagglutinin head domain, which is exposed. So all these changes, these dots, what you are seeing, these are the changes are occurring in hemagglutinin. So hemagglutinin, you cannot use it, but if you look at the stem of hemagglutinin, that is not that many changes. So many people think about that when a broadly protected vaccine is going to be used, probably hemagglutinin, the stem region can be used, or the M2 protein, which is a channel protein, and then the other one is NP, and some can also be using some internal protein. So we started with HA2 and M2E, and Immunogen make a lot of construct and did the challenge study and all that, and we find it out that this type of vaccine can provide cross protection, but protection is partial. So we thought probably we'll be better off moving to another antigen. So we started looking at nucleoprotein, and the main reason for nucleoprotein that with influenza A and influenza B, this virus is more than, or this particular antigen is more than 90% conjured. So the chances of getting a vaccine which can provide protection, which is NP based, then this will be a good idea. So in addition to that, what we add in our vaccine, there is a small peptide, 21 amino acid long, and that is autophagy inducing peptide. So what it does, that if you attach to your antigen, it will take through autophagy pathway. So antigen presentation is not going through that pathway, but now people understand that if your antigen go through autophagy pathway, they have a different mechanism of antigen presentation, so you can enhance the antigen presentation. So when we use this antigen when we use this peptide, then with the RNA-seq, with the different, you can say, confocal microscopy, RT-PCR, some inhibition study, all that we try to find out that antigen presentation, if we use this particular peptide, it will go through this galactin and cathepsin dependent pathway. And with that, we also saw with some antigen, the T-cell response is significantly enhanced. So our vaccines, these are mostly the T-cell based vaccines. So in there, what we did, that we created vaccine with influenza A, from A virus, NP, then from influenza B, and then one vaccine we combined together, and we created in the bovine adenovirus format and also in the human adenovirus format. So immunogenicity study, we did this one single dose of either bovine, human, just wanted to see that which one is going to do better. Then did the humoral immune response and the cellular immune response. And then later on protection study where we did only with bovine, and that was also a single inoculation because no two inoculation. So when we are looking at the serum, looking all different type of immunoglobulin, IgG, IgG1, G2, and IgA. Since it's serum, IgA in serum are going to be less. And these inoculation are intranasal. So upper panel is for influenza A virus, and lower panel is for influenza B. So influenza B response is usually going to be lower. So wherever we have, say, influenza A NP, then in immune response is higher against influenza A, and when we have influenza B, then influenza B. In one thing, we have influenza A plus influenza B, and there we will get immune response against both of them. So in here, it seems like both the vector are doing well when we look for even lung washes, and lung washes are going to have similar immune response with its humoral immune response will be better IgA response is going to be there, but overall lower influenza B response, and that is always known that influenza B, even with the hemagglutinin, the response is going to be lower. So then we look for the interferon gamma secreting CD4 T cells by flow cytometry, and also the interferon gamma CD8 T cells. And this one we use the spleen for that, also used in this, you can say, left-hand panel is influenza A virus and or influenza A NP, and the right is influenza B. So influenza B, and in both, the bovine adenovirus is doing a better response in both of them. And then we look for by ELISpot assay, looking for interferon gamma excreting cells, and these are in the number. So in splenocyte, middle triangle lymph node, and in lung mononuclear cells. So higher level are going to be in middle triangle lymph node and in lung, because we are doing intranasal inoculation. And overall, it seems like the bovine is doing slightly better in T cell response. Here we did not compare with or without our that magic peptide, because we have done multiple study, and every study the peptide is providing better T cell response than without peptide. So that is for influenza B, a similar result, but a little bit lower exposure. Then we also look for some tetramer assays. So we designed some tetramer, and the company provided us those tetramer, and to find out those CD8 T cells will be going to be effective or not, and find it out that with influenza A, influenza B, they will be pretty good overall. So whenever we do an immune response, if we don't get protection, then all that kind of thing is not that useful. So what we did in this one, when we challenged with them the regular virus which causes seasonal flu. So the influenza A1, H1N1, then H3N2, and then 2 influenza B, the Yamagata lineage, and also the Victoria lineage. So when we look at all these data, where we are looking for the virus in the lung, so wherever we have influenza A and P, there is a lower titer in the lung. Where I have influenza B, then there is a lower titer. And in one we have combined, then in both of them is lower in there. So consider this one is combined, this one is combined. So this is the vaccine we probably are going to propose. And when we have these 3 viruses which are potentially pandemic virus for human, so H5, H7, and H9, and look at in this vaccine. So the amount of the virus, it is much lower. And when we look at some viruses which we can use in mice, where we are looking for the mortality and morbidity, and our vaccine is providing protection from mortality and morbidity against both of these viruses. So we look for H1N1 because we can do that study, H3N2, and we can also do with the Yamagata strain because the other one does not kill mice. So basically there was no more morbidity. If you look at this red arrow here and here, and if you look for the specific one, these one they will be with individual one. So either influenza A or influenza B. So many times we also like to do another species. So ferret is considered a animal where the influenza naturally can occur in ferret, and many virus can replicate in ferret. So we did our vaccine only the bivalent vaccine in ferret. So we look for the immune response. There are not that many reagents available for ferret. So we look for the IgG response with prime and boost, and we find out in the boosting there is a huge immune response. We also look for T cell response using the peptide library, and there was T cell response which is very specific for influenza A. H1N1 was there, then response against H1N1, then H3N2, and then with the influenza B. And similarly, also in splenocyte and also in the blood mononuclear cells. So we also looked at the temperature is going to occur or not. So in the control, at least in H1N1 and H3N2, there was a day where they have the temperature, but in the vaccine, we did not get any temperature. But if you look at all these three here, that this is control, this is the vaccine one, this is control, this is vaccine, this is control, this is vaccine. So it seems like that using these three different viruses in ferret, it seems like that the vaccine which is containing both NP from influenza A virus, NP influenza B virus is providing protection against both influenza A and influenza B viruses. So this is the summary that a single dose will providing T cell response and the B cell response, and that T and B cell response is providing protection against a number of influenza A viruses and influenza B viruses. And seems like using influenza A and influenza B nucleoprotein, that can be considered for doing more work to have a broadly protected influenza vaccine candidate. So putting that one, you can say just in the picture-wise, that in the control, that was another study where we did 2 doses. So consider when we have all these influenza A viruses, so wherever we see mortality, these will be there, then they have a huge amount of virus in the lung. But when they are protection, then we are not seeing anything in all those animals. And this particular work was, you can say, done by Ekrami, who is here, and he will be starting a faculty position in our lab pretty soon. So these are actually that work I presented. You can say majority of that work was done by Wenxing Wang, a graduate student in my lab. I always has collaboration and help from CDC, and over there we have about 20 years collaboration with Dr. Sambara, and the funding is from NIH. And that's all for today, and I'll be happy to answer any question you may have.
Questions, comments, please.
Yeah.
I think this was fabulous, and I thought it's really impressive how a single dose can drastically reduce your titers to the levels that you just showed. I also think it's quite impressive that you're getting both a T cell and a B cell mediated response. But mechanistically, what I don't fully comprehend is why your CD8 positive response is so sustained. I would expect it to kind of come up first and then go down as you continue with your CD4 positive and the IgG titers. But you're seeing a sustained CD8 response. So the reason I'm asking is, like, mechanistically, it's interesting because if that's simply a function of the AIP being used, then it's something that could be applied to several other antigens to kind of maintain
Yeah.
that response.
So in this one, we did not did the mechanistic study, and I think a lot of work needed to be done to finish that mechanistic study, because first thing is, what are the correlate of protection? How the protection is attained? It is B cell protection or T cell or a combination of both. So what we believe, probably it's a combination of both. So then how NP antibodies are going to provide protection? So more likely that protection there are in literature is ADCC based protection. It is not neutralizing antibodies. So those adaptive transfer with the cells, and then also the cells you collect which are important, which are in the lung, rather than cells which are in the spleen. So which type of the cells? So you can say first question, are the T cell response can last longer? In our system, we have done a study where we did for 1 year. So 1 year, we did not find any decrease in T cell response. Up to 1 year, it sustained. And when we do the challenge, so since we saw that much T cell in those mice, and when we challenged with that H5N1 that was done in BSL-3, and these mice were fully protected. So seems like that system, the T cell response can be durable. So currently we will be starting that study soon, which will be done for year study. So we'll see this vaccine, how durable immunity they are providing, and are these any type of immune response is going to last for a month, 2 months, or about a year. So that will be a year-long study, and hopefully we'll get some interesting outcome out of that study.
Can I ask you a related question?
Mm-hmm.
With your sustained T cell response, have you ever tried re-challenging the system?
Say it again.
Have you tried re-challenging the system with another viral infection?
So re-challenging, I think re-challenging will be much better, because the immune response after challenge, it is going to enhance all these immune response, and also to some of the other thing like HA, we did not have HA, so there will be immune response against HA and NA as well. So I think if we re-challenge them, the protection is going to be much better.
Or re-challenge with other strains.
Yeah, other strain, yeah.
Variants.
Yeah,
To
That
prove
would
that
be,
it is universal.
then
Yeah.
it would work.
yeah.
He means that heterologous protection. So our NB is from H7, and we are challenging with H1, H3, H5, H9.
Yeah.
So it protects. So the heterologous protection is there, because this protein is maybe the same in all of these, but the natural infection is not providing enough immunity against this protein.
Yeah, because we have just NP from just one virus. Yeah.
Dr. Mittal again, thank you very much. Interesting talk. As you were talking about the CD8 and sustained immunity over a period of time, if you think about the life of a mouse versus life of a human, right? You have longer, thankfully, a longer lifespan. So, question for you is, do you suspect that this sustained induction or stimulation of immunity can have other consequences, such as neoplasia or T cell tumors of some sort or anything like that, because of constant induction? As you know, even with a lot of tumors, the first step is inflammation. If you look at cats with IBD, followed by a leukemia. So do you worry about any kind of like a secondary, tertiary impact of this?
So, not really, because if those things occur with inflammation, and in some organ you find some sign are there, then it will be a worrisome sign. But when there is all the organs are just fine, and you get the immune response which is there. So since the antigen stimulus is gone, the antigen stimulus is not there, because the virus will be there in the body for a week or so. After that, the virus is gone. So how it is being sustained is those memory response is that much, because many times you consider that it will go down. Because when we were looking at the B cell response, means antibody, hemagglutinin antibodies and other antibody, they were getting down with the time, but they were still there after a year. So with that, we thought, if it is that way for mouse, it is wonderful. But in the end, we have to have a vaccine for a target. In this one is human. So until we go up to there, otherwise, if a vaccine is for ferret or for mouse, we have the vaccine. But is this same vaccine is going to work for mouse, or it is going to work even in cattle? So the cattle study we will start this year to find out that, and we have a different vaccine construct for cattle. So is it going to work in cattle or not? And is the vaccine or the antibodies are going to come in the milk, where the majority of the virus is there? So we are at the planning stage, but the mouse study for the cattle vaccine, mouse study we can start next month. But the cattle study currently we have planned in June. So when the weather is better for those animals, because the colder climate, the vaccine effect can be a little bit different. You had another question?
Yeah, I was just wondering if you ever looked at your IgM titers. Because if your IgM titers stay sustained high, that explains the memory part of it and why you were getting such a strong, effective neutralization.
So we have not looked at it. So we have those serum sample. We can go back and can look at it, and that can answer the question Dr. Nayan is asking. Yeah.
Hey, Suresh, thank you very much for your presentation. I was just impressed by the losses in the bird flocks, and just wondering whether that your vaccine might be protective for birds against avian flu.
So at that study, we are currently pursuing with Ohio State, because they have the BSL-3 facility. So they will be better off doing those study. So those study we have just started, and we'll see what will be the outcome of those studies.
So we've got time for one more question.
Yeah.
Do you know anything about the epitopes you're seeing, how many epitopes and what epitopes you're seeing the TCRs respond to in the NP, and where do they fall in the protein, if so?
So one protein we were looking at, that protein falls at, the number is 147, and that is, you can say, very strong T cell response. We also find it out when we did those antigen library, so which are overlapping. So we also found there are some other one, and we used more of this one because regularly majority of the people in influenza virus, they are using this 147 peptide for T cell response. But I would say there will be at least 3-4 which are significant T cell epitope for influenza, especially in NP.
Well, thank you again, Suresh. Fascinating, timely presentation.
Thank you.